Welcome back to our virtual Friday field days. This week we're going to do something a little different than we could do at a normal field day, and we are going to take you behind the scenes at the grains lab. Once our samples have been processed for test weight, grain moisture, and germination, we bring them back to the grinding room. We process a lot of different kinds of grains here. This is a commercial wheat sample, a commercial rye sample, some wholeless oats, and some of our winter barley malting trial. We write down the analyses that need to be performed and the sample number on the jar, and then we put them through the laboratory mill. We always want to grind about 300 grams so that we get a good representative subsample. We have that marked on the hopper, and then suction pulls it through the hammer mill, which is not turned on for this demonstration or plugged in, and then it goes through a sieve. A lot of our machinery is calibrated to specific grains and specific particle sizes, so we want to make sure that we've got a really nice uniform particle size, which is why we ask our commercial folks to send in whole grain samples that we grind on site. This is how we measure crude protein and flour moisture. We scoop the sample in, we tamp it down in with this special little press, and then what the machine is doing is called near infrared spectroscopy. And so it's shooting little beams of light at this sample. And then from how it bounces back, it can tell us, calibrated for that particular grain, what the moisture is in the flour and what the protein level is. And so for this barley sample, it's got a 15.1% moisture content and a 9.1% protein. You may have seen the starch in the window. We do not have it calibrated for starch. So that's the cell that it's packed into that Rory is cleaning out right now. And that little window is where the light streams come out and also where they're measured when they bounce back. And the machine can determine the composition from that. We use the flour moisture to help us set up our sample size for our next test, which is falling number. You might remember falling number from when Henry talked about our rye harvest date trials. So falling number measures how long it takes a weighted disc to fall through a test tube as that test tube is being heated with a mixture of flour and water inside. This is a proxy for the enzymatic activity in the grain. Grain that is not mature enough has a really low falling number. That weighted disc basically falls straight through the tube. Around 250 seconds is your best, and then if the grain is over mature and it starts to pre-germinate, you'll get a high falling number. So we grind it up, we weigh out the sample, we add 25 mils of water, we shake it up to get it all in suspension, and then it cooks inside the falling number machine and gives us our falling number. So this is the test tube and the weighted disc, and what happens as it's cooking is it slowly drops through that tube, and when it hits the bottom, there's a sensor that sees that it's there, and there's your falling number. And Rory's going to show us how it's actually done. We add the 25 mils of water. We get a nice tight cork and shake it all up. If there's flour stuck to the bottom, it can incorrectly read that as already having fallen down the bottom. So we always make sure it's completely in suspension. And then we make sure we scrape down the sides with the disc and make sure that all of the flour is in the tube. The tube goes into a little plastic collar that fits over the top of the machine. If you drop it in without the collar, you make a big floury mess inside your falling number machine. And it grabs a hold of that plunger on the top and rapidly shakes it up and down for the first minute while it's starting to cook. And this is what it looks like when it's done. This is a test tube that's completely cooked and the weighted disc has fallen to the bottom, and that's how we measure falling number. So one of the most common tests that we run in here is testing for a toxin produced by a fungus, which is called Deoxynovalenol. And the FDA limits this toxin for human consumption at 1 part per million in the finished food. So we have to make sure that we are being really accurate and precise with this test, since we're measuring parts per million level. So we always measure out exactly 10 grams of flour. We add that to an Erlenmeyer flask. We add exactly 100 milliliters of water. And like with the falling number, we shake it up to make sure it's in suspension and not stuck to the bottom, and then we put it on the shaker for 3 minutes. Once it's finished shaking, we let it settle out for a few minutes, and then we filter out the flour, and what we're measuring is the filtrate that's left behind. We combine that with some chemicals, and this is what we call a colorimetric test. So one of the reactions that's happening in there with one of the chemicals, it produces a blue toxin if the a blue color if the DON is not present. If the DON is present, it reacts with that one substrate, then it can't bind to the other molecule that produces that blue color. So if you see that blue, you know that you don't have very much DON present, and it goes through to a pink color, which is what you get if you have a lot of DON present. We haven't seen much of this toxin this year because it's been a nice warm year. We don't just look at this ourselves, though. We put it through a machine that shines light through it and reads the exact wavelength, so it can tell us exactly how blue this is versus pink, where the light falls along that wavelength, and then it correlates that back to the specific level of the toxin. So we put it in the reader and we press start. There's a couple of ways that we check ourselves against this. One is that every time we run this standard curve with known amounts of the toxin, and the machine calculates an R-squared and lets us know how close we are. We also participate in a proficiency testing program from the Neogen company, where quarterly we run samples with a known amount of the toxin, or an unknown. They know how much of the toxin is there. We also test our own research samples for forages and corn. These go through a separate mill. It grinds things finer, and we have it completely enclosed because it's grinding a finer particle size. Also because a lot more of these things, particularly the forages, contain things that can be irritants. We grind these up at the research farm through a Wiley mill, so they're already partially ground when we get them. And then we put them through this mill and grind it to a very fine powdery particle size. And that's how fine the powder is when it comes out of the forage mill. And then we run them through another NIR, which is again using that near infrared light to determine the composition of these samples. This is a much more generalized one. You can run corn on it or forages or silages, but you don't have to tell it the specific species. It uses these cups with glass bottoms. They're all chipped, so you can tell if you have it in the right way, and it moves them around and scans them with the near infrared streams of light, and then can tell from the spectra that's produced what the composition is. So for these, we have to fill this cup up. It doesn't have to be completely full, but the bottom has to be completely covered. We spread it around a little, and then we tamp it down with this lid, making really sure not to actually press on it, just kind of letting it set down on there to even things out. If you compress it, it changes the readings. And it reads through the bottom of that glass cup. We put it in. We press the button, and then the magic happens. The lasers are going. We have to tell it what the sample number is, and it gives us a pretty detailed report afterwards of all the different components that make up the forage or the corn sample. It tells us the dry matter, it tells us the protein, it tells us how much digestible and non-digestible fiber are in it. It can tell us the chemical composition as far as individual elements that we might want to know what the nutritional quality is. And it also tells us how digestible that particular forage is. So thanks for joining us in the Grains Lab.